marker. My name is Kazuhiko. I retired from the University of Tokyo March this year and took the position of the professor at the Tokyo University, Kumamoto, Japan. So I know how hot and how humid they are. I was a cowboy in the United States as a Japanese agricultural trainee. This is 40 years ago, and where only 3 nights a scene at night in Camas County, Idaho, USA. And my life was not miserable. In fact, I had a great time there because I watched the cattle every day. And these observations became the foundation of my research career. And this is 40 years ago. You see the gap between the now and 40 years ago. I was a cowboy. And this is only 1 year ago, and this is my favorite horse. And many times I need to demonstrate how to ride a horse at the University of Tokyo farm. And now you may think that I was able to ride a horse long time ago. No, yes, this is only 5 months ago. I was demonstrating how to ride a horse. Now, this shows the changes in pregnancy rate of cattle in Japan and for the last 25 years. And the Department of Agriculture says that we need to have the efficient reproduction system in Japan. And as estimate, the 10% decline means result in about 50 million, not yen, $50 million loss a year. This is a huge loss in Japan. And this is pregnancy rate of the artificial insemination and embryo transfer program in Japan. And usually the fertilization rate is 95%, but yet when we determine the pregnancy rate exactly, about day 30 or later, the pregnancy rate becomes like only 40 to 55%. If in fact the sperm, egg, fertilization, or like a blastocyst formation are the problem for this low pregnancy rate, then we should be able to overcome this with the in vitro fertilization and embryo transfer. Because fertilization rate is 100%, blastocyst formation is 100%. Yet, pregnancy rate is somewhat between the 45 to 65%. It's slightly better, but not as much. So I'm not saying here that the ovulation, sperm, egg, fertilization are not important. They are important, but processes and the cellular mechanisms associated with implantation are very important because this is the time where a lot of the early embryonic losses occur. And this simply shows one way to improve pregnancy rate is just to make sure that we reduce early pregnancy loss around here. This shows day 12 blastocyst, epiblast, Hatch blastocyst, and this the uterine lumen, epithelium, and glandular epithelia. And then this is elongating conceptus on day 16. This is 1 to 1.5 days after the initiation of conceptus elongation, and about 2 days after maternal recognition of pregnancy. This is the elongating conceptus, luminal epithelia. This is a glandular epithelia. And this is really elongated conceptus, and this is day 20 pregnancy, and glandular epithelia. And conceptus attachment in our system begins on the day 19 to 19.5. This is so day or half a day after the attachment is initiated. And please note that still you can see the distinct epithelial cells. And this is day 22, pregnant uterus. And the epithelia is not as distinct anymore. And this is more like, and this is a trophoblast right here, and uterine epithelium right here, stroma, glandular epithelia, and not as distinct luminal epithelia. So events, this summarizes more or less the events associated with Conceptus implantation. Of course, you need to have the great maternal recognition pregnancy, so resulting in the maintenance of corpus luteum and high progesterone secretion. And during this time, Conceptus migrates, hatches at positions where the attachment occurred. Then implantation wind, how Conceptus is allowed to begin its attachment. And immunological adjustment. And we have detected, not only we, but all others detected that the immune cells are really accumulated at subepithelial regions. And as Conceptus start to elongate, the binucleate cell begin to appear. And those binucleate cells start to produce pregnancy-specific hormones. And of course, if this is the more invasive, you need to have invasive control on the invasion, and then finally the placenta formation. But point is, this is where like a 50% of losses occur. So today, I'd like to share with our data with you that during this time, Conceptus or uterus use the molecules associated with the lymphocyte homing, and I'll explain this one a little later. And here, adhesion, epithelial mesenchyme transition. This is commonly seen in cancer metastasis. And in a sense that this is more like attachment, and this is more like to separate. And this two different opposite things happen at the implantation site. This is a lymphocyte homing. This is just a blood vessel. And lymphocyte, and this need to slow down and transmigrate into the affected regions. So it uses cell adhesion molecules such as L-selectin and these guys and the VCAM. And today I will show you only this data and this data. This is a regulation of the endometrial L-selectin expression. And when we culture a luminal epithelium and we add like this, the day 17 pregnant, 20 pregnant, day 20 pregnant, uterine flushing on top of that one, then we can enhance the L-selectin expression. This is a transcript. And also the constituents of the uterine flushing, EGF, a basic FGF, when we added this one too, we can increase the expression of L-selectin, and this is a protein. So messenger RNA and proteins. And this is VCAM expression in utero. VCAM is expressed both this is the elongated Conceptus right here. This is luminal epithelia, elongated Conceptus. VCAM expression is seen at both luminal epithelia and elongated Conceptus, trophectoderm. However, the receptor to VCAM is integrin alpha 4, which is expressed only by the Conceptus. And this again shows the in a dose dependent manner that these addition of FGF and EGF, FGF could increase VCAM expression. Then likewise, proteins are also increased. enhanced by the addition of growth factor or interferon tau. This is the epithelial mesenchyme transition, commonly seen in cancer metastasis and organ development. Basically, these the cancer cells are epithelium, and epithelium usually stays tightly bound and stays. But when this becomes the mesenchymal in nature, this goes through like lymph duct or blood vessels to go to different place, different organs, and make epithelium to mesenchyme to epithelial transition and become the epithelium. Then that increases as the cancer cells. And these are transcription factors commonly seen during the epithelial mesenchyme transition. And this is also seen in the Conceptus attachment. This is day 22, bovine pregnancy. This is elongated Conceptus right here, luminal epithelia, and this is enlarged portion. And cytokeratin is a marker for epithelial, and vimentin is the mesenchymal marker. And as you can see, that Conceptus, the trophectoderm, is epithelium, and the luminal epithelia also stained for cytokeratin. So they are epithelium cells. But when we look at the vimentin, of course, the luminal epithelia is not stained, but elongated Conceptus is stained with the vimentin. So the morphologically, elongated Conceptus is not only respond to the epithelial marker, but also mesenchymal marker. Now, the function is, E-cadherin is the functional marker for epithelium, and N-cadherin is a mesenchymal marker. And as you can see here, that elongated trophoblast was not stained for E-cadherin. Of course, luminal epithelia is stained for E-cadherin. However, when we look at N-cadherin, this is again functional marker for mesenchymal cells. Then these are stained. So a very kind of interesting thing. So cytokeratin plus, vimentin plus, E-cadherin minus, and N-cadherin plus, the situation. And to begin, we suspected that EMT should occur like before attachment process is initiated. But yet, as I indicated that in our case, Conceptus attachment begins on day 19 or 19.5. And yet, EMT occurred only day 22. We don't have the data on day 21. Cows are very expensive in Japan, but we do have data on day 20. And day 20, no EMT. And day 22, EMT. And we don't have we do have day 25, but we don't have day 23. And we have, many times we stopped collecting samples on day 22 in the morning because we could still perform the uterine flushing. But afternoon of the day 22, we are no longer able to collect much of the uterine flushing media. So many events are in progress concomitantly during the implantation, such as Conceptus growth and development, and Conceptus uterus communication, receptivity. And this is a point I really would like to share with you today, is that Conceptus implantation while epithelial mesenchyme transition proceeds. So in a sense, there are two opposite events, one to attach and other to separate. And trophoblast and embryo cell differentiation, and of course placenta formation. But this is the point of the talk today. And other thing is that how can we improve or stop the pregnancy loss or improve pregnancy rate? Then we thought that not each event is important, each molecule is important, but not each event or molecule. Understanding of the uterine constituents as a whole or as a sequential events, as an implantation process. We need to understand those. Unless we understand those, we may not be able to increase or improve pregnancy rate. So we started to characterize uterine flushing media. And this is a strategy to characterize uterine environment. Of course, we have done next generation sequencer or microarray or 2D gel. But in this case, we opted for the eye-track analysis. This is a comprehensive protein analysis and a semi-quantitative. And after we identify some candidates, then we developed the co-culture system where we have epithelial cells at the bottom, and then we add the bovine trophoblast cells, CT1 cells on top of it. And then we can manipulate or we can overexpress the candidate proteins so that we can really determine which minimum number of the proteins that could mimic uterine environment or to improve the uterine environment for better pregnancy or less embryonic loss. And at this point, this is the uterine flushing media, so we expected that it's going to be like maybe 100, maybe 200, maybe 300 at most. Yet when we executed this one, we found 1933 proteins and in uterine flushing, and on day 17 in 2022, interferon tau is present. And when we look at interferon tau, this is a comparison between this and this. So interferon tau decreased, so we know the system is working. And we also noticed that there are so many TGF-beta related genes are upregulated and downregulated. And using the bioinformatics and other systems, we reduced the number of the candidates from 1,933 to 213. Then we started to really examine each of those candidates using the co-culture system with uterine epithelial cells and trophoblast cells. My student did, not me. And this is a candidate from the eye-track analysis, a TJ-related gene expression. And as you can see, that follistatin is highly expressed on day 20 and low expression on day 22. The red one is the Conceptus, and the blue one is endometrium, pregnant endometrium and non-pregnant endometrium. And some others are expressed in endometrium, but these guys, vasopressin, and these guys are expressed highly in Conceptus. And we then examined in different way that on the uterine flushing, on if we can detect the follistatin, and this is day 17 pregnant, day 20 pregnant, day 22 pregnant. And we also did immunohistochemistry and highly expressed in the elongated Conceptus. This is where attachment started, after attachment started. And then we look at the inhibin isoforms. Then we found that on day 22, activin A is highly expressed. Day 20 is not. And we were wondering why this happened. And we figured out that follistatin is inhibiting. I will show it later, but the point is, we found increases in endometrial activin A on day 20. A slight increase, but then further increase on day 22. And using, again, the co-culture system, this is the epithelial cells usually, and the trophoblast on top, the spheroid trophoblast. And we try to mimic if the EMT-associated transcription factors are regulated or not. This is the SNE1, SNE2, ZEV1, and TWIST1. And as you can see, that if we add to this culture the day-28 flushing media or activin A itself, we could increase its expression, EMT-related transcription factor expression. And when we use co-culture system, this is now uterine epithelium here, and trophoblast on top, we can really mimic that expression of these similar to EMT with only by using the activin A only. So in this case, only for these EMT markers, again, SNE2, ZEV1, and TWIST1, activin A alone was as effective regulator as uterine flushes. And we are continuing this kind of the analysis to reduce the number of candidates so that minimum number of the proteins could be applied to improve uterine environment for a better pregnancy rate. And this is a recent analysis that transcription factor OVL2 was expressed high on day 20, but reduced on day 22. This is a mRNA, and the protein is similar way. On day 20, the highly trophoblast is expressing the OVL2 highly. And we did quantitative PCR and a microarray analysis, but this is the one I really like to share with you, that this, using the, again, co-culture system, we allowed the trophoblast attachment to the uterine epithelium or not allowing attachment. And when we allowed Conceptus attachment to the uterine epithelium, we could reduce OVL2 expression, and we could increase others. Again, these are EMT-related transcription factors. So this summarizes all of my talk in a sense, that using the L-selectin or VCAM, these are all found in the lymphocyte homing, and these are more like required for the attachment. And yet, on day 19.5 or day 20, follistatin expression or OVL2 expressions are very high, that inhibiting the activin A or these transcription factor related expression. However, when follistatin and OVL2 decreases, and for that decrease, attachment is required, then the break is gone, then all the EMT-related transcription factors are upregulated and really become the EMT-like. And we believe that both the attachment and the EMT, again, to separate, is both two different things occurring, because this adhesion, we think this is more like attachment of these cell types through the cell adhesion molecules. This is attachment, more like adhesion between the trophoblast cells and the uterine epithelial cells. And this adhesion between the trophoblast and the uterine epithelial cells result in the trophoblast flexible to form the fused cell. Around this time, we see some of the binucleate cell formation, but at this time, we start to see the trinucleate cell formation. And for most of the mammals, the binucleate or trinucleate or multinucleate cell formation is always at the toroblast side. And in ruminant, it has been believed that trinucleate cells are the result of a fusion between the binucleate toroblast and one of the epithelial cells become trinucleate, end up the uterine stromal cells. Whether or not this is true or not, we could see still the trinuclear cell in the stroma. And we could see the binuclear cells as they start to elongate. But no one has demonstrated, actually actively demonstrated the formation of trinuclear cells. I've seen this one in the stroma, but not the process itself. But point is, these two different things are occurring, and it is required for the toroblast cells to become epithelial cells, to become flexible, to form the fused cells. And we have worked on the horses too, and horses are similarly oriented. Horses Conceptus, it doesn't elongate, but similarly oriented, really close by, but still has there some space in between the toroblast layer and epithelial cells. And that may be the kind of common system for so-called non-invasive mode of the implantation. And this, we wrote this one earlier last year, to the Journal of Endocrinology. And that time, impact factor was 4.7. And in 2017, this was rated, our review paper was rated 5th, not the first one, 5th of the most downloaded a year from the Journal of Endocrinology. Now, to summarize, we have a lot of time, but to summarize how we can improve pregnancy rate. We can improve uterine environment. We need to improve uterine environment, allowing cell adhesion molecule expression, and at the same time, epithelial mesenchyme transition to proceed. So we need to identify the minimum numbers of the uterine factors and develop the way to use these guys as a substrate to increase the pregnancy rate. We have been working on it, and from the 1933, we reduced number of candidates to 8 right now. Once we reduce it to 4 or 5, we start to develop the kind of substrate to apply for the actual situation. And we also developing the way to find the cows not pregnant, and probably, hopefully, we could determine that one on day 20, 19, 20, or 21 after artificial insemination. And this kind of stuff is not only us. Pennsylvania State University, Troy Art is also working on it. I know what he's working on it, and we talk a lot. And he has been using the ISG15, interferon similar gene 15, and MX1 and MX2, and the combination of those, he thinks that he can determine the cows not pregnant on day 20. We have worked on that one. However, in our system, those ISG15 is there, but MX1 and MX2 are not our candidates. But he thinks that he could determine the cows not pregnant on day 20, 20 days after the artificial insemination. Although he indicated that he needs to increase the number of experiments. And this is my last one. And I have collaborated a lot with the people at U.S. Department of Agriculture, Pittsburgh State, and everyone. This is Korea. He always, when he introduced me in Korea, he always said, everyone, this is my brother stuff. And These are all the university Tokyo, all PhD students. And hopefully, I moved to the Tokai University last April, and we have many names here to continue our productive laboratory. Thank you very much.